Dr. Karl Deisseroth of Stanford, your work exemplifies many of the examples of cool science that are happening here at this conference. Could you describe it briefly?
Yes, what I have developed and work on is something called optogenetics. This is the use of light to control brain cells, and it's something that we can do in a very specific, speedy way, millisecond precision, and targeting just precise cells and precise connections within the brain. And this has allowed us to understand what the cells are actually doing, what their causal impact is on behavior, even in the complex mammalian brain.
So you can actually turn the circuits off and on and see what that does.
We can turn them off, we can turn them on, and we can do it in animals that are expressing psychiatric disease-related symptoms like anxiety or drug addiction and substance dependence.
In fact, you just had a major paper came out about anxiety.
That's right. We published a paper on anxiety in Nature. The first author was Kay Tai. It was from my laboratory, and we had a series of experiments in that paper that found that there was an anti-anxiety pathway within the amygdala, which is a brain region that had largely been thought to be involved in fear and anxiety promotion, and we found quite the opposite, buried deep within it, an anti-anxiety pathway that we were only able to resolve with optogenetics.
So that's tremendously appealing that we might be able to turn off our anxieties with light. What are the implications for potential human treatment?
Well, the most important implication is simply on understanding, on showing that there are specific cells and specific projections within the brain that can serve to either promote or inhibit psychiatric disease symptoms. For many people, that's an incredibly important insight, just to know that there's a real biological basis and a circuit basis to these disorders. It helps us understand them. It'll help us treat them by whatever means, whether it's through drugs or brain stimulation treatments.
So we could actually use light on human brains?
We could. More in the near future, and perhaps more broadly, the use of light in animal models will help us develop other kinds of treatments for people that will give us better and more safe and effective therapies.
Okay, but potentially we could insert the needed genes eventually in human brains.
Yeah, there's no particular barrier to that. One would have to be very cautious, of course, thinking about possible immune reactions and the like, but it is perfectly possible.
Great, thank you very much.